What is biopharmaceuticals? Any drug product manufactured in, extracted from, or semi-synthesized from biological sources. At the start, you need to know that there are a number of ways in which such drugs can be made, but the key distinction between them and the other drugs is that they are not extracted from a native source or synthesized with chemical reactions, and instead they are created with the use of living organisms which may have been modified to produce the desired compound. They can be also classified into three classes: drugs extracted from living systems, produced by recombining DNA, vaccines, and gene therapy. How are biopharmaceuticals made? A container used to create tightly controlled conditions which facilitate the growth of a particular organism. Drugs can be produced into it by organisms which generate biopharmaceuticals as a byproduct of their life cycle, often because these organisms have been modified to produce specific proteins and nucleic acids. Cell cultures and modified microbes can both be used in bioreactors to make drugs and compounds, which can be used in the production of pharmaceuticals. Genetic modification of plants and animals may also be used to make biopharmaceuticals. And now to the production steps. Production of a biopharmaceutical involves many different, complex, and lengthy steps. We may divide them into 4 steps. The first one is synthesis. The second one is purification. The third one is formulation. And the last one is final dosage form preparation. At the first step, which is synthesis step, we order the raw materials needed to make the product and test them to be sure they meet quality standards. For biomanufacturing, equipment and materials need to be sterilized to avoid bacterial and other contamination to the cell cultures. The product is referred to as the active ingredient. The original cell cultures is started in small bottles, around the size of large soda bottles. As the cells grow and multiply, they are introduced into a small bioreactor. Eventually, they are grown in large bioreactors, which can be several stories tall. Then after the active ingredient is synthesized, it must be purified. So here comes the second step, which is purification. It involves removing the chemicals used in the process. It also involves separating the cells from the cellular nutrients and byproducts they grew in. The end result of production is called the bulk product. It may be sold as it is, processed further at the same plant, or shipped to another plant for further processing. Several other operations are required to get bulk product into its final form. This takes us to the formulation step, which implies chemical mixing operations to blend the active ingredient with other substances, such as fillers, needed in the final form. Biopharmaceuticals usually are sold as sterile liquids or sterile powders. Finally, we have the final dosage form preparation step, through which the formulated preparation is made into its final form. The final form is dispensed into containers, and the containers are labeled and packaged. When researchers develop new biopharmaceuticals, they typically file for patents to protect their inventions and the process, and go through a series of steps to seek approval so that their drugs can be sold on the open market. These steps involve extensive testing for safety and efficiency to confirm that the drugs work as claimed. So as you can see, it may take more than 15 years to develop a single drug.